Hi students, here we are going to discuss two topics in the sexual reproduction of the flowering plants. These two topics are most important for the Neet exam, remember it. And he didn't ask the question, only one question came from the double fertilization after 2020. But before 2020, he asked 4 times about the double fertilization, triple fusion, but he didn't ask this one. So chances are more in the 2026 Neet. Questions will come from this. Now we'll see artificial hybridization. What is artificial hybridization? Already you know the cross pollination, how the cross pollination will be taken place. That previous video I discussed. And in the cross pollination will be done in between the two known varieties which are having good characters. Which are the what is good character? What are the good characters, desired characters? Here it is, for example, generally it is to take place in nowadays in the two varieties. Means variety means what? Under the species, so many varieties used to present. So in between the two varieties of a species, it will be done. And the two varieties used to have some good characters. They will select how it is. For example, if you take the one plant is one variety plant is having high disease resistant and drought resistant. Second one is having high yield but less disease resistant and less drought resistance. So if the both the characters are coming on one plant, then it will get the more yield and high disease resistance also, highly economic to the farmer. Such type of things will be done, and we are getting the hybrid seeds nowadays. Everyone knows what is hybrid seed. That will be formed by the artificial hybridization, because this itself also can come. The concept of this in the Neet question. Now here they will take, that is the process, and here you see what they are doing. Artificial hybridization, it is one of the major approach of crop improvement program, because nowadays we are getting high yields compared to last 40 years. Before 40 years we are not getting this much yield because of this artificial hybridization only. In this one, so it has taken, it is a major approach of crop improvement. Means crop improvement means yield improvement program. Artificial pollination made between pollen grains and desired stigma. Already I told. That is desired means good characters, whatever we want. We want means desired means good characters containing pollen grain, one plant from the one variety, and next is the desired stigma where it is present in the female plant. So that female plant are in the, that is the part of this pistil. So desired pistil from one plant and desired anthers means pollen grains from one plant, both are having different desired characters. We are doing this one to bring those good characters in one plant. That only we are the major approach of the crop improvement program. How they will do? Because if the plant is, if the female plant parent as hermaphrodite, I already discussed in the previous video, that is 90% of the plants are hermaphrodite, means bisexual flowers they used to produce. Bisexual flowers are there, that the outbreeding devices I discussed thoroughly in the last video. So in that one, chances are more to the autogamy, because stamens are there, pistil is there, autogamy can be taken. So in that one, what we will do, we will remove the stamens in the female parent. What we selected female parent, out of the desired characters are present, one we will select as a male parent and one will be the female parent. So if the female parent used to be the hermaphrodite bisexual flower, so we have to remove the stamens in the early stage before dehiscency, means before releasing the pollen grains, we have to remove it. That removing of the stamens before maturity in the female parent is called as emasculation. This is most important one. Removing of stamens before the dehiscency in the female parent if it is hermaphrodite. These are all things, whatever words I discussed, it is a very, very important, remember it. And this is the emasculation. How they will do emasculation? Emasculation will be done like this. Here, in the bud stage, they will open with two forceps, and they will open the bud, and keeping it on the plant only. They will open the bud and removes the stamens. Here is the scissors, but with the in the textbook he has given forceps. So we will remove anyhow. And after removing this one, what we will do? So after removing this one bud, we will cover it with a bag. That is butter paper bag. Butter paper, why? Because it is the aeration will be there for that flower. So butter paper bag, we will cover it. After removing the stamens, that is process is called as emasculation. And after bagging this one, they used to observe every day. Whenever this whatever the pistil ripen, the stigma is ready to receptivity, it will be taking proper pollen grain. They will collect from the male plant the whatever the desired pollen grain and open that bag and dusted that pollen grain on the stigma. So this is the desired pollen grains are kept on the desired female plant. So naturally this pistil must accept this pollen grain. Why we are keeping the bagging? Bagging is not there. Unwanted pollen grain also will come and fall. But we are doing the experiment, so it should not be. The unwanted pollen grain should not reach on the stigma of wanted female parent. So we used to protect it by the bagging. And after when the stigma is ready for the receptivity, means matured and it is ready to take the pollen grain, we will collect the pollen grain from the male parent and open the bag and dust it on the stigma of a desired flower. And again we will bag it, because after putting the pollen grain also, it may be contaminated by other pollen grains, means other varieties or other something will be happen. So to avoid unwanted pollen grains to reach on the stigma, bagging is required. What is bagging, he will ask. What is emasculation, he will ask. So bagging will be done. Bagging won't be done on the male plant. Why, he will ask? Because pollen grains let come and reach on this one. We don't want. We want only pollen grains from this one, male plant. So bagging is not required. Now, whenever we are seeing here, so after bagging, after dusting of the desired pollen grain on the stigma of the female plant, we will keep it for the up to the formation of the fruit. So the fruit is formed, the seeds are hybrid seed. Desired male plant and desired female plant, we done the cross pollination. This is called as artificial hybridization. Now you see here, it is one of major approach of crop improvement program. Okay, artificial pollination made between desired pollen grains and desired stigma. The female parent is hermaphrodite with bisexual flowers. Stamens are removed in the flower. Stamens are removed in the bud stage by emasculation with a pair of forceps. With a pair of forceps, stamens are after emasculation. The bud covered with a butter paper bag to avoid unwanted pollen contamination. I already discussed. When desired stigma reached receptivity, desired pollen dusted and rebag. Again, we used to rebag it because to avoid the contamination. Until fruit formation. If the flower are unisexual, bagging is not required. Emasculation is not required. Unisexual flower means female parent, you will have the only pistil. Female flower will have the only pistil. So it is not necessary for the emasculation, because if it is having the bisexual flower, then only emasculation. If it is a unisexual, not necessary to the emasculation. Just we have to bag it, because it should not be contamination. And desired pollen grain, we used to dust, everything is common. But it is important point given in the textbook also. Emasculation, bagging, and what is desired character, what is the object of this artificial hybridization. All these things combining, he can ask a question. I am expecting a question in the Neet 2026 from this. And next, whenever we are seeing double fertilization, here double fertilization, we will see the diagram. Here it is, already we discussed this one, how the pollen pistil interaction means pollen is to reach on the stigma, and it will germinate and forms the pollen tube, and pollen tube will grow to the style and reaches to the ovary, and finally it will reaches to the ovule that he has shown. I discussed in that video. And now what happens after reaching into the, after the pollen tube is reached, by the time of the it will have two male gametes. Generative cell used to divide and forms two male gametes, or right from the beginning it may be having the two male gametes. But with two male gametes, pollen tube used to reach and enter into the ovule. And ovule, after ovule, it will enter into the pollen embryo sac. Embryo sac contains of 7 cells, 8 nuclei. These three are the egg apparatus. Central cell contains 2 nuclei and antibodies 3 cells. Now, by entering into the embryo sac, what it will do? The pollen tube will enter into the one of the synergid. Important point. It used to enter into the one of the two synergids are one of the synergid, and it will be guided to enter into the synergid through filiform apparatus which are present at the polar side in the pore, germ pore side. That is it is called as a micropylar end of the synergid used to have the filiform apparatus. It will guide the pollen tube to reach into the synergid. Remember it. The pollen tube is entering into the synergid, not into the entire. Only one synergid, even though it is having two. And then it will rupture and it will release both the male gametes into the synergid, and the synergid will be ruptured, and it will send one of the male gamete into the egg cell, and other gamete will be sent to the polar nuclei. So one of the male gamete will fertilize the egg, which is received by the synergid through the pollen tube, and one will be fertilize the egg. Second male gamete will fertilize the double nuclei. So this is the first fertilization will be done with egg. First fertilization will be done with egg, and this is called as one egg fertilized by the one male gamete. It is called as syngamy. It is called as a syngamy. First fertilization is over, and one more two male gametes came. One more male gamete will go, and it will come and fertilizes the double nuclei. Already it is having the two polar nuclei. One more male gamete is coming with one nuclei. So all the nucleus of male gamete and both the polar nuclei, all three will be fused. Anyhow, this male gamete is fertilizing the double nuclei. So it is called as second fertilization. So double fertilization, one fertilization with egg, second fertilization with the polar nuclei. So in the polar nuclei, whenever it is fertilizing, triploid nucleus is to form, because already it is having n number, 2n number. It is haploid, two haploid nuclei in central cell. These two haploid polar nuclei will fuse one more haploid nucleus of the male gamete. So it will become triploid. So this is called a triple fusion. Fusion of one haploid male gamete and two haploid nuclei of central cell. So it is called as a triple fusion. We used to call. So double fertilization, triple fusion. Fertilization will be taken place generally one only. But here it is taking two fertilizations. It is a main characteristic of all the angiosperms. In the angiosperms only it used to happen. Double fertilization, triple fusion is a unique characteristic of angiosperms. This we will see in the matter. Now you see here, double fertilization. Pollen tube reaches pollen tube releases two male gametes in the cytoplasm of synergid. One of male gamete fuses with egg along with nucleus and nuclear fusion along with nuclear fusion fuses and formed by the syngamy forms the zygote. That is one of the male gamete used to go and fertilizes the egg. It is called as syngamy. I only wrote the thing, but because of this, both these very new to me, some problems are coming. Anyhow, I think you can understand. So next is the second male gamete fuses with dinucleate central cell, because I just already shown two nuclei are there, haploid nuclei cell, and forms triploid nucleus. Two haploid nuclei of central cell and one more haploid nucleus of the male gamete will be fused together and forms the triploid nucleus, which is called as primary second endosperm nucleus. The triploid nucleus, now it is called as primary endosperm nucleus. And syngamy, zygote will form. Male and female are mixing, means egg and one male gamete is fusing, that is syngamy. That is called zygote. But here triple fusion is called as a triple fusion, one nucleus is formed. That is called as a primary endosperm nucleus, the most important bit. And now, after triple fusion, the entire central cell is called as a primary endosperm cell. Primary endosperm cell. Remember, primary endosperm nuclei by triple fusion used to form. Up to that time, we are calling the central cell with the double nuclei. Now the entire central cell is called as primary endosperm cell. Remember the point. And because of the second male gamete fertilizes in secondary cell, it is called double fertilization, and formation of primary endosperm nucleus is triple fusion. Double fertilization, triple fusion we used to call actually. This is what is double fertilization. Two male gametes will reach from the pollen tube into the synergid, and one of the male gamete will fertilize by syngamy with egg, and the second male gamete will fertilize with the central cell, which are having the two nuclei, haploid nuclei. So one haploid nuclei used to come through the male gamete, and this male gamete nucleus and the two nuclei which are present in the central cell used to fuse together and forms triploid cell, triploid nucleus. The triploid nucleus is the triploid formation of the triploid nucleus is called triple fusion, and two male gametes are fertilizing in the two places is called double fertilization. And whatever the triploid nucleus is formed, that formed triploid nucleus, now it is called as primary endosperm nucleus. And after the double fertilization, the central cell is called as a primary endosperm cell. This is the double fertilization triple fusion, and forms a pen into the triploid. Now secondary cell called primary endosperm cell. Now I am asking one question here. Remember it. This is most important. Try to answer it and discuss with your lecturer. From a endosperm cells, a scientist developed a plant in tissue culture. Tissue culture means we will take some tissue and we will form the new cell. That is tissue culture, you know. What is the scientist done? He taken some cells from the endosperm and developed into the plant, and in the tissue culture, this plant can do sexual reproduction. I am asking you. He will give so many options. He may give something. Try to answer it. Think over it. Okay, I am telling it cannot do sexual reproduction because in the meiosis has to be done. Meiosis cannot be done with the triploid cell. Triploid, three sets of the chromosomes. Diploid cell only, diploid nuclei only it can do. Triploid will fail. Why it is? Think over it. Now once more we'll see here. These are the two topics: artificial hybridization and double fertilization. These are the things which I shown. This is the artificial hybridization with emasculation. He has to do because it is a bisexual flower. If it is a unisexual flower, emasculation is not necessary. And in the bud stage only it will be removed the stamens and bag. We used to close it with the butter paper bag to eliminate, to avoid the unwanted pollen grains. So it should not be contaminated with unwanted pollen grains. And next, that whenever the pestle used to reach maturity, the desired whatever the pollen grain will be dusted on that one and it will be rebagged, and the rebagging will kept up to the fruit formation. The removing of the stamens in the bisexual flower, female plant means we are selected as a female plant, so it is bisexual flower, we have to remove it. That is called as emasculation. Emasculation is not necessary if the unisexual flowers are there. If you are doing the experiment in the unisexual hybridization, it is not necessary. And double fertilization, triple fusion, when the pollen tube is entering into the embryo sac, it will bring two male gametes, and the two male gametes will be released into the one of the synergid, and the two male gametes, one of the male gamete will fertilize the egg, second male gamete will be fertilize the secondary cell nuclei, polar nuclei. So one of the male gamete fertilizing, fusing with the egg cell, it is called as syngamy. Male and female gametes are fusing is called as syngamy, and zygote will form. The second male gamete will fuse with the two haploid nuclei which are present in the central cell. So the two nuclei, haploid nuclei, polar nuclei of central cell, and one more nucleus which came through the male gamete. All three nuclei used to fuse and forms the triploid nuclei. Triploid nucleus. So that's the one fertilization with the egg, second fertilization with the central cell haploid 2 nuclei. So it is called double fertilization. And the second fertilization, it is forming the triploid nucleus. So it is called the triple fusion. And now the central cell is called as, after fertilization, second fertilization, the central cell is called as primary endosperm cell. And the triploid nucleus is called as primary endosperm nucleus. These are the things. If you like this video, please give the like mark and subscribe the channel, because I want to complete this. I am completing already plant physiology I completed, but in the writing on the writing board. And now I started this sexual reproduction on the digital board. Within 2 days I will complete this chapter. If you want to see those things, please see by subscribing the channel. And the next topic I am going to start principles of inheritance, followed by the molecular basis of inheritance. Very, very important topics. So subscribe the channel and see all the things. One thing I can say, I am confident that each and every video, my video, is having something new points which are not covered by the books also. I am damn sure. I am having 32 years experience. And I used to see all the things. But where they are failing to the representation, those things I cut, and I given all the things which are important for the Neet. And secondary for the 12th class students who came to the 12th class, they also are getting the name Neet type of the questions in the 16 bits he used to give in the starting. So it is required. Please follow my videos, then you will get the concept and the Neet orientation, how the twisted questions can be formed. Okay, thank you.